Oh, okay, got it. All right, so let's all welcome Dr. Marie Simonin, who will be giving us our guest lecture today. She's very well versed in microbiomes and largely studies plant microbiome interactions and how disturbances such as climate change or pathogens affect microbes and ecosystems. Since receiving her PhD from University Claude Bernard and University Joseph Fourier in France, she has completed postdocs both in the US and in France. She is currently a researcher with INRAE, INRAE, which is France's National Institute for Agriculture, Food and Environment, where she's part of the Research Institute in Horticulture and Seeds Lab as part of the EMERSIS system team. That was weird. Anyway, she has been awarded multiple grants, and her current research includes studying the impact of seed microbiota on seedling emergence and also the use of pesticides on seeds. Marie's done a lot of great work in the microbial ecology field and has published several articles. In addition to this, she's been involved in the scientific community by reviewing articles and grants, giving a lot of presentations in various locations, and being an educator and acting as a mentor and supervisor for both PhD and master students. It's a pleasure having you here, Marie, and we look forward to hearing about your research.
Oh, thanks a lot for this very kind introduction, Sarah. It's really nice. And so today I'm going to present some work from my postdoc in the US on the impact of anthropogenic disturbances on stream microbial communities. Especially we were looking for microbial indicator taxa of those disturbances. And so feel free to ask questions or interrupt me if you want. But just as a quick introduction about my research interest, basically I'm trying to understand what's the ecology of the Anthropocene. And so I'm really interested in the impact of different types of disturbances on biodiversity and especially their impact on environmental microbiomes. So my work is really at the interface between three different disciplines. So ecology, global change science, and ecotoxicology. And I've been doing my research on a great diversity of disturbances, including pollution, pathogens, climate change, land use change, and so on. And also on a diversity of ecosystems, including soils, grasslands, wetlands, rivers, and so on. So today I'm going to focus on my aquatic work, but feel free to ask questions on other ecosystems. And so the way I see this research is really, we can, at the interface between these three disciplines, it can be considered as a discipline called stress ecology or disturbance ecology. And so today I'm going to present two case studies. So from my time at Duke University in Emily Bernard's lab, during which I'm going to show some results on the impact of urbanization on river microbiomes and our approach to identify microbial indicator taxa. And a second example on the impact of mountaintop coal mining on aquatic biodiversity as a whole, not only microbes, using an eDNA approach. And so this first case study on the impact of urbanization, well, the approach we used was amplicon sequencing, targeting the 16S gene. So maybe you've heard of this before, but we aim to characterize the sediment bacterial community using this amplicon sequencing approach. So just on bacteria here. And we took advantage of a large urbanization gradient that exists in North Carolina, close to Duke University, that is really well characterized. So we collected sediment samples from 41 rivers that really cover, we can say, forested watersheds or reference watershed to highly urbanized watersheds in the rivers. And so our question in the along this urbanization gradient was, can we identify microbial indicator taxa of urbanization? And this question is like, or basic one is just to understand which microbes can be sensitive or resistant to urbanization. But in general, we think that microbes can be a very interesting tool for biomonitoring. And usually we use microinvertebrates, insects, or fish as indicator taxa, but actually microbes can be very sensitive to change in the environment. And also sequencing now and those approaches of amplicon sequencing are actually cheap. So it's actually cheaper to analyze microbial communities than insects, macroinvertebrates in the field. And so our approach was to use classic gradient analysis in ecology that have been applied to plant communities or insects and so on, to understand the response of organisms to variation in the environment. So to do this, we used an R package called Titan. It's a method that is called a threshold indicator taxa analysis, but also quadratic regressions. The idea is that you have an environmental gradient here, and you're going to look at how the abundance of the different taxa will vary along this gradient. And what we did was to identify which taxa were responding negatively to this gradient, or positively, or were not responding. But also with quadratic regressions, we were able to identify what we call subsidy stress responders. So meaning you see that they peak somewhere along this gradient. So a little bit of urbanization is good, but not so much. So subsidy meaning that they benefit from the gradient that maybe provides some nutrient or optimal niche for them, and then it becomes too stressful. But the problem to perform this analysis was that urbanization, it's a complex problem. It's a multiple stressor problem. You have a mix of disturbances that happen in the same location. So you have a nutrient pollution gradient, a cocktail of inorganic and organic contaminants like metals or pesticides. And you also have huge physical disturbances, including storms, light pollution, noise pollution, and so on. So our question was, okay, we want to do gradient analysis on the distribution of the bacterial taxa along in our samples, but what here is our x-axis. So what is a good proxy or a good variable to characterize this very complex environmental gradient? So our approach was not to choose one, but to actually run the indicator taxa analysis on all the environmental variables that were correlated to the bacterial community structure. So I'm going to show you the results. So here we are representing a multivariate analysis. So maybe you've seen this before as a PCA or PCOA. Here it's an NMDS, where we looked at the structure of the microbial community. So each point represents a sample, a microbiome, if you want, and the closer they are, the more similar they are in terms of bacterial community. And here, each point is colored by the percentage of development. So it's a land use proxy or measurement that we have for each river. And so at 0, it's a low urbanization, and red is high urbanization. And you can see that there is a shift in community structure. You have more like the forested or undeveloped rivers, and here you have the one that have the most urbanization. And we've measured tons of variables on those streams, including physical and chemical and land use and so on. But we found that 4 variables were really correlated to the distribution of the bacterial community. And they were the percentage of forested land in the watershed, the percentage of developed land, and the concentration of zinc in the sediment, and a biotic index. So the biotic index is a stream environmental quality index that we measured based on microinvertebrates diversity. So it's what is used classically to assess the water quality in a stream. And so we use then for a Titan analysis, so the threshold indicator taxa analysis, those 4 different environmental variables to identify our indicator taxa. And so here I'm showing you the tree of all the taxa, the bacterial taxa we observed in 40 rivers. And so here you have each branch represents a taxon, and here are the different phyla or classes. And I'm going to show you on this tree, which taxa were found to be negative responders to urbanization in blue or positive responders in red. And they have different symbols depending on if they were identified based on biotic index, development, for, percent forested, and so on. So what you can see here is that we found 138 negative responders to urbanization, and they were really distributed everywhere on the bacterial phylogeny. So really, urbanization is a huge impact for bacterial communities, and proteobacteria, or acidobacteria, bacteroidetes, and so on, bacteria that are phylogenetically extremely different were identified to be negatively impacted by urbanization. But interestingly, we found some conserved response. So some taxa were more negatively impacted than others, especially here, the Xanthobacteriaceae, and especially the genus Varibacter. They represented 39% of in this group, 39% of the OTUs or taxa were found to be negative responders. And here in the family Acidobacteriaceae, it's 50% of the taxa that were found to be negative responders. So those groups, and you see they have multiple points, so they're responding to multiple different variables. So they're potentially robust indicator taxa. Now I'm showing you in red the positive responders to urbanization. You see that they are also distributed along the phylogeny, but there are not that many. We found 56 taxa. Some group like Acidobacteria, we never found a positive responder there. But we mainly found them in the beta proteobacteria here. So the Comamonadaceae, for instance, and mainly among proteobacteria. So this is just the approach we used, and then from this we can get a list of potentially interesting indicator taxa, both positive or negative responders. And we also looked at the subsidy stress responders, so that have a peak along this gradient. So you see again that they are really distributed everywhere on the tree. So the subsidy stress response is a very common one among bacteria. So they can be a little bit tolerant to urbanization, but often then too much urbanization is not good for their survival. And just to show you an example of the typical distribution of a subsidy responder, if we look at this guy here, you see they are basically absent or very rare at low development and also at very high development, but they peak in the middle of the gradient. And if we do this analysis for all the different taxa, subsidy responders, stress responders, we found that they peak usually around 40% of development. So really they peak at intermediate level of urbanization. And that's interesting too. And that can also help us understand more about the ecology of all these different bacteria. And so right now I showed you results only at the taxa level. But what is interesting with Titan is that we can use all those taxa level responses, so all those aggregate the results of all the sensitive or tolerant taxa and calculate their tipping points. So for each bacteria, we can know where is their change point along the gradient, their tipping point, and aggregate all these results to identify community level tipping points. So really to understand at which point along this gradient, where we can observe a synchronous decline in the diversity or the abundance of the bacterial community. And so this is really interesting from an application or biomonitoring point of view, because then we can say, well, have an idea, say, okay, it's at 20% development that we really see a large impact on the bacterial diversity. And this can give guidance, for regulation and biodiversity protection. And so what we found with these results is, like, when we look just at the sensitive taxa, we found that their tipping point at the community level was around 12% of watershed development. It's actually very low. It means that they are quite sensitive to urbanization. And if we look from the different point of view, from the forested land, so if the change point is around 50% of unforested land, so, and the sediment is 13 microgram per gram of zinc. So just to give you an example of potential application and information we can get from this taxa and community level analysis. So in conclusion for this first case study, using this classic gradient ecology approach, and we applied it to microbiome data, which is not necessarily very commonly done. We were able to gather new knowledge on the ecology of these microorganisms from a fundamental point of view. So to know more about their habitat preference, their tolerance or resistance to disturbance like urbanization, and potentially understand more about their life history strategies and the habitat that are adapted for their survival. From a more applied point of view, we are able to identify indicator taxa, the proportion of sensitive and tolerant taxa in a community. And that could be a useful information to understand the future stability of those microbiomes to future disturbances or an intensification of urbanization. And potentially perform microbiome engineering to restore some of those communities. And so an interesting thing is that from this taxa level information, we can then infer community level tipping points that could be useful for environmental monitoring and pollution regulation. So that was the first part. Now I'm going to show you another example of this type of approach, and focusing on the impact of mountaintop coal mining on their impact on aquatic biodiversity using an eDNA approach, so eDNA for environmental DNA. And this work was recently published in Ecological Applications. So just to give you a brief introduction on what is mountaintop coal mining, basically the idea is that you have mountain with coal. Instead of digging a tunnel like the traditional way, you're going to just explode the entire mountain and remove it and fill the valley with all the debris and the rocks. And so you can directly access the coal seam. So you can imagine this is a huge environmental impact. And so basically the landscape is becoming flat after this. The mountains are disappearing. And it actually represents 40% of coal production worldwide. Especially it's performed in Central Appalachia in the US. And most of the coal mines are there. And we know that there is a huge impact on water quality, of course, from this. And especially in this area in West Virginia, 10% of the mountains are mined. So it's like a huge impact on the landscape. It's not like 5, 6 mountains, it's like 10%. So it's a very significant land use change in this area. And one of the signature of the impact of coal mining in general is we see a strong correlation between the percentage of the watershed that is mined and the water conductivity. So the salinity of the water, basically. And a typical water conductivity in a reference stream would be close to 100 microsiemens per centimeter. And in mined area, it's often higher than 1,000. So what this conductivity means for, what does that mean? It's a high level of mixture of salts, mainly calcium, magnesium, sulfate, carbonates, and nitrates, so that leaching from the exploded rocks, basically, that have been displaced from the top of the mountain to the valleys. And this is called alkaline mine drainage, because here in this case, this high level of salts also lead to an increase in the pH of the water. It's not acid mine drainage, it's alkaline mine drainage. And we also observe a huge nitrogen export, 10 to 60 times higher than in reference streams. So this is well known that there's a huge impact on water quality. So there's been some previous work to make some recommendation to protect aquatic life in those streams. So from the EPA, the Environmental Protection Agency in the US, that to try to protect aquatic life, we should aim to be below 300 microsiemens per centimeter in terms of conductivity. And those are the streams we sampled. So you see, it's not really well respected so far. We found plenty of streams that have really high conductivity. And so this aquatic life benchmark or criteria that was established by the EPA is based on macroinvertebrate data, so classically used in biomonitoring. Basically, this is the exposure level at which 5% of the macroinvertebrates genera are lost from the streams. The thing you need to know about Appalachia, it's like a huge biodiversity hotspot in America. And those mountains are among the richest in temperate areas, and there are more fish species in Tennessee than in the entire European continent. And this is a huge migration corridor for birds, mammals, and amphibians. So there are really plenty of interest in understanding what really is the impact of mountaintop mining on biodiversity as a whole, not only on microinvertebrates, but previous biodiversity assessment, they were only focusing on macroinvertebrates and fish communities. But here our objective was really to perform a regional assessment on the impact of mountaintop mining on riverine biodiversity, but really across the tree of life, so from bacteria to fish. And to do this, we used environmental DNA. And so from the same DNA, we can get all the information from a water sample. We can get all the information on all the biodiversity present in those streams, from bacteria to vertebrates. And the idea was to, again, identify taxa and community tipping points to increasing stream conductivity in those streams, and compare what we found. So by not only looking at macroinvertebrates, but all the organisms to see how their sensitivity compared to existing water quality criteria, like the 300 microsiemens per centimeter EPA one, and see if this is actually protective of all the organisms or not. And so to do this, we collected water samples from 93 streams in West Virginia. And we collected from streams that were the same amount above or below the EPA criteria at 300 microsiemens per centimeter. And so eDNA, its collection is really easy. So you just collect a water sample, it's like 200 milliliters with a sterile syringe and filter. And at the same time, we were also doing water quality assessment using YSI probes, and we looked at the different, at the chemistry of the stream, the organic carbon, and so on, and different metals. And so you can see from the picture, it's really pretty there, but sometimes you work at the confluence between a mine stream and a reference stream, and you can easily see that those rivers are very different. And so then we extracted the DNA from the filters here, and we performed amplicon sequencing using the Illumina MiSeq technology. And we targeted different genes to target different groups of organisms. So 16S for bacteria, 23S for algae, 18S for all the other eukaryotes, not including algae. But also we targeted specific groups of organisms to have a higher resolution in the taxonomic assessment. So for macroinvertebrates and for fish. And so to the results, not surprisingly, we found a very strong impact of mining on aquatic biodiversity. So here you see a similar representation as I showed you before, looking at the community structure of the bacterial community. So the points are colored based on water conductivity. So the red one, they are basically in streams that have mining activities, and the white one are reference streams, if you want. So you see clearly a separation in the communities. And this is even stronger for algae. You see really a group of points that are clustering together in the unmined streams and all the other mined streams separating. And for eukaryotes, it's the same thing. So clearly a large restructuration in the community structure of bacteria, algae, or eukaryotes in general. And in terms of species diversity, so the number of taxa in those different groups, so that we can assess based on the OTU richness, so it's the observed bacterial richness here, you see a trend for a decrease in the number of species identified along the water conductivity gradient. And for bacteria, you see it's most points are below the reference, the average diversity in reference streams, but the pattern is really clear for algae. It's like a 50% loss. And similarly for the general eukaryotes. So you see that it's not only a recomposition, restructuration of the community, it's mainly driven by a loss of diversity. And so we found that those declines in species richness, they averaged, it's about 50% loss of diversity for those different groups. If we now look at specific groups or the macroinvertebrates, so those results are still preliminary because the primers, the targeted genes, it's still mainly under development to have a good accuracy in the taxonomic identification and resolution. But here you see all the different species that we identified and in which the number of sites in which they were detected. So the white one are the one below 300, so unmined, and the red one are the mined streams. And you see that we detect those macroinvertebrates most frequently in unmined or low conductivity streams. And we have a low detection of all those different taxa in mined streams. And for the community structure, similarly, we see a separation or a shift along the conductivity gradient and a large loss of richness for macroinvertebrates. And for fish, the detection was a little bit more difficult. We were not able to detect or amplify fish DNA from all the streams, but for the ones we were able to do it, you see it was mainly from streams that are unmined or below the 300 microsiemens. And so a very low detection of fish in mined streams in red. So now we use the Titan approach to characterize the individual taxa responses to the conductivity gradient. And you see here the percentage and number of taxa that were found to be either negative or positive responders to mining. And what you see here, you have the percentage is we have 12 to 25% of the taxa for the different groups that were identified as sensitive to an increase in water conductivity. And that most of the taxa, when they were significantly affected by this gradient, were negative responders. We have few positive responders to this increase in conductivity, except among bacteria. And if you look at those results on a phylogenetic tree, so here you have the tree for algae, again in blue, the negative responders, in red, the positive one. A striking result was like most diatoms, so Bacillariophyta, were found to be sensitive. It's not surprising, but we confirmed what is previously known from the sensitivity of those groups to environmental disturbances. And we have very, very few positive responders in the algal community. For bacteria, we see sensitive bacterial taxa in all phyla. They are everywhere, there are tons of them. You see all the blue points. But we found also, again, some positive responders, like urbanization, mainly among proteobacteria. And for the eukaryotes, the sensitive taxa were mainly fungal. So here, especially the Chytridiomycota. And we found also some protists, Kinetoplastida or arthropods, or even the plant phyla that were affected by these mining activities. if we aggregate all this information to the community level to look at community tipping points using Titan, you see that we calculate those community thresholds for the different groups and compare it to the EPA reference at 300. What we found it was that generally the bacteria, algae, or macroinvertebrate groups presented community tipping points closer to 150 or 170 microsiemens per centimeter. And there is a huge variance, but they are generally have a lower threshold than 300. But for the eukaryotes, it seems that this is actually protective for this group, if we look at general eukaryotes. But if we split them based on different groups, it's actually lower than the 300 criteria. So in conclusion, we found that mountaintop coal mining is leading to large shifts and declines in aquatic biodiversity, not only of macroinvertebrates or fish, but generally across the tree of life. Using eDNA, we were able to find declines in aquatic biodiversity close to 40 or 50% loss of taxa, depending on the group considered. And we found large synchronous declines in community. So it's not just one group that is sensitive, it's generally the entire community that is affected. And that happens at very low level of mining. So 10 or 15% of the watershed that is mined is enough to cause already large impact on the stream biodiversity, corresponding to conductivity values close to 150 or 200 microsiemens per centimeter. And so I wanted just to highlight the promising applications of eDNA. So eDNA is what we've been doing to study microbial communities for a very long time, because we didn't have an alternative. We need to extract the DNA and sequence it to characterize the community. But so far it was not done for larger organisms. You can go on the field and collect plants, fish, and insects. So now we are proposing to transpose this approach used for microbes for also larger organisms, and to really target all kingdoms of life in a single sample, with single water samples. And in this assay, so on almost 93 streams, we're able to detect 10,000 different taxa. So it's a very sensitive approach. And we can use it to have both information at the taxa level, but also at the community level to set up or revise existing environmental regulations. And this is a water example, but you can also have the same approach for soil biodiversity, for instance, on microfauna to microorganisms. With this, I want to thank you for your attention, and all my colleagues from Duke University. Just to let you know, we are recruiting 2 master's students next year, if you're interested. It's not on the aquatic ecology, it's on microbiomes, more plant microbiomes and applications in agriculture, but feel free to contact us if you have questions. And I would be happy to address all the questions in the chat.